Yeah. Now, the chemical reactions and enzymes. Last time we took the chemical reaction and we understood the parts of chemical reactions. We said that we have the reactants and products. And then we explained how the energy is released or absorbed during the chemical reaction, and then explained or discussed how the solutions or how the reaction could be exothermic or endothermic. Then we explain the activation energy. Who remember what is the activation energy? Raise your hand if you want to answer. Who remember what is the activation energy? Activation energy. Abdullah Malik, yes.
The amount of energy required to start a reaction,
The amount of energy. Okay.
required to start an action or like reaction.
Excellent. Thank you. Let's see. Yes, yeah, Mustafa Khan. Yes, Mustafa Khan. Okay, thank you. Yes, Muhammad Abu Ghandar.
It is the energy that is needed to cause the reaction between the reactants forming the product.
Okay. Mustafa said can't. Yes, now you can, Mustafa. Yes.
So it's an energy that's activation energy is the energy that starts up and chemical reaction, a
Yes.
small energy that starts up.
Thank you all. So it's the minimum amount of energy that is required to start the energy, right? And we said that this needed in exothermic or endothermic reaction, but the activation energy, of course, will be bigger if the reaction is endothermic or exothermic. Hands. Yes, Tijan.
Usually it's exothermic.
Yeah, in exothermic,
Sorry,
the activation, endothermic or exo?
Yeah, endothermic.
Endo. So the endothermic reactions usually have a higher activation energy, and we took these two graphs last time. It's important to understand it and to understand how to point the activation energy. We said in the exo or the energy absorbing reaction, sorry, the energy absorbing means that the products will carry higher energy than the reactants. The activation energy, it's the hit point to the reactants. Okay, this is the activation energy. While here, for example, we have exothermic reaction. The reactants carry energy more than the products, means that the energy is released. Or exothermic, the activation energy, it's between the reactant and the top of the energy level. Okay, so always the activation energy is it's between the reactant energy level and the top energy level. Okay, this is the activation energy, reactant energy level, the top energy level. This is the activation energy. Enzymes. What are the enzymes? You know, in chemistry we have something called catalysts. Catalysts are chemicals or substances that speed up the reactions. But how it speeds up? We said last time that the reactions, chemical reactions, have different time or different, let's say, rates. We call it rate. That the time, just to understand, when we say rate word, rate means, or reaction rate, it's the time which is required to convert all the reactants into products. Again, the reaction rate means the time which is required to convert all the reactants into products. Got it? Okay. This is called the reaction rate. Now, enzymes speed up the reaction. What does it mean speed up the reaction? It minimizes the time which is needed to convert the reactants into products. Now, sometimes some reactions need hours or days to convert all the reactants into products. Enzymes or catalysts in general, catalysts, minimize the time which is needed to convert all the reactants into products, okay? Or we can say speed up the reaction, speed up the reaction, or minimize the time which is required to convert all the reactants into products. Is it clear for all? If not, open your mic and speak. Good. Now, I don't know if you can open your mic or not, but let me check. Okay, now you're allowed to unmute yourself. Abdullah Malik, can you repeat? Okay, Abdullah Malik and others. Just a second. Where's the whiteboard? Why it's switched off? Just a second, guys. I want to open the whiteboard too.
Mr.
Yes, Habib.
Excuse me again. What is the name of the time taken for the reaction? Like you said the name.
Wait, yeah.
Something.
I'm coming. Wait, I will repeat. Just I'm opening the whiteboard to share it with you and tell you everything. But give me a second. A second one. Okay, now I can share it with you. Yes, look here. Now, the reaction rate, the rate of reactions. Okay, the rate of reaction. What does mean the rate of reaction? The rate of reaction means the time I don't know what's wrong with the whiteboard. Yeah, the rate of reaction, it's the time that required, or we can say the time that is needed to change all of the reactants into products. Now, if we put two chemicals together as a reactants, these chemicals and these chemicals, okay? These chemicals will react together to become a product. So here we have the reactants, and here we have the products, okay? Now the product supposed to be, let's say like that, okay? These are the products. So converting all of the reactants into products, this is the rate of reaction. This is what? The rate of reaction. Got it? Now, or the time which is needed to change all the reactants into products. I think is clear for all. Now, some reactions, they have long rate. Some reactions have long rate, okay? Others, they have short rate of reaction. Means that longer time or shorter time to convert all the reactants into products. That's it. Okay, what are the catalyst does? Catalysts speed up the reaction. What does mean speed up the reaction? Means minimize or reduce the time that needed to change the reactants into products, all the reactants into products, okay? That's what we mean by speed up the reaction. Again, the catalyst is a chemical we added to the reactants. Okay, it's different than the reactants, and the catalyst does not react with the reactants. Okay, does not react with them. It just provide a suitable place or suitable condition to speed up this reaction, speed up this reaction. Okay, such as provide a good temperature or provide a good pH for the reactants to react together very fast. Okay, now in biology or in the living cells, we have a biological catalyst. Biological catalyst. This biological catalyst called enzyme. The cell make this biological catalyst to speed up the reactions in the cell. This enzyme, by the way, all enzymes, let's take some features about these enzymes. All enzymes are made of
Proteins.
excellent proteins. Number one, enzymes are not used up in the reaction. Are not used up in the reaction means that do not share with the reactants or do not react with the reactants. It just provide good place, good condition, okay, for the reactants to do the reaction faster. How it speed up the reaction, how the enzymes speed up the reaction. Look at this, very important. Enzymes lowers the activation energy. The activation energy. Now we know that reactions need activation energy, or reactants need activation energy to change to product. Okay, some reactions stay for hours or days just to gain the activation energy to become product or to react and become product. The enzymes lower this activation energy. Instead of gaining much of energy to start the reaction, the enzymes make the reactants start the reaction with less activation energy. Got it? So here, if I draw a graph, we have reactants and products. And as we said, we draw it like that. These are the products. These are the reactants. Okay, here the energy level. Okay, now how much activation energy needed to do this reaction? If it's from this point to this point, from the reactants to the highest point, you remember we said this. This is the activation energy, right? The activation energy. Okay, if you add enzyme, this is the activation energy for this reaction. If you add enzyme to these reactants, enzymes lower the activation energy. It becomes like that, for example. Okay, so in this case, sorry, just let me do something because the line not clear, I think. Yeah. Now, look. So the blue line represents the reaction with enzymes. Okay, the red one without enzyme, without enzyme. So that means that the reactants will react together in very fast way rather than waiting to get or to gain much of activation energy to start. So less activation energy are good or useful to start this reaction. Is it clear?
Yes.
Is it clear for all?
But Mr.
Yes.
don't you
Yes,
need extra
clear.
energy but Mr.
Yes.
Why does it lower the activation energy? Doesn't more energy helps more, like into breaking and causing a new product easier?
Yeah, this is the idea. Now, sometimes the reactants need, need the activation energy to change its shape so that they can react together. The enzymes provide a good suitable place for the reactants. Look at this. For example, this is enzyme. Let's draw it like that, for example. Okay. These are the reactants. The reactants bind on a binding site on the enzyme. This is the enzyme, and these are the reactants. Okay. The reactants can bind on this side, which we called it what? Binding. Yeah, binding site. Okay. The binding site, or sometimes they call it the active site. Okay. Here, it binds here on the binding site. So that provide a suitable place so the reactants come closer to each other and easy to activate, to be activated and react together. Okay. So here,
Yes.
the enzymes provide a suitable place. But if you notice here, if a reactant such as like this one cannot bind on the active site. Why cannot bind on the active site? Because the shape is different than the active or the binding site shape. Okay? So
Yes.
that lead us to add another point for the enzymes, that enzymes are specific. Enzymes are specific. Means that the enzyme which used, for example, the enzyme which used to break down maltose into glucose and the glucose. You remember? Glucose and glucose give you maltose, right? Yeah, and here plus
Yes.
H2O, water. You need water for hydrolysis reaction. Okay? So water and maltose react together to give you glucose and glucose. This enzyme, or the enzyme which required here, is specific for maltose because the maltose is the substrate, we called it. Substrate is the molecule which is fit with the binding site, or the molecules which can bind on the binding site of, or the active site of the enzyme. Got it?
Yep.
Yeah. So
Yeah.
every enzyme is specific for a substrate. Maltose, the maltose substrate is specific for specific enzymes. So that's why scientists call the enzymes by adding ASE to the end of the substrate. Yani this enzyme, we called it maltase. Maltase. Adding ASE at the end. Maltase is the enzyme of a breakdown maltose. Okay, so what should we call the enzyme which is required to break down galactose?
Cheese.
Huh?
Lactase.
By the way, what are the
Lactase.
products? Tell me first what are the products? Galactose. Break it down into.
Glucose
Mr. Isn't
fructose.
that lactose
Yeah,
broken to
answer my question only. Huh? What we call the products? What are the products of galactose breakdown?
Glucose and fructose.
Yeah, glucose plus fructose. You sure?
Uh,
Open your
Glucose
mic and
plus
speak.
water? Glucose plus water?
No,
Uh,
this is
Mr. Lactose.
disaccharide. Disaccharide react with water. Supposed to give you monosaccharides. What are them? Glucose and
Lactose.
lactose. MashaAllah. No one noticed the mistake, huh? You sure? You sure?
Mr. Isn't lactose made out of lactose and glucose?
Bonus 2 for you, Muhammad. Thank you. Only Muhammad noticed this. This is monosaccharide. The disaccharide is lactose. But no one noticed that. Come on. It's lactose here. And here. galactose. Excellent, Muhammad. Thank you for this.
Okay.
Thank you for your attention, Muhammad. You see, the lactose is the disaccharide. I just said the galactose disaccharide. No one told me no, mister. Galactose is monosaccharide. Okay? Lactose is the disaccharide, which we break it down by hydrolysis reaction into monosaccharides, glucose and galactose. Okay? What we call this enzyme, which is required for this reaction?
Lactase.
Lactase. Lactase. Just add ASE. Who can give me example? Raise your hand. I will choose names. Who can give me examples of another disaccharide or polysaccharide? You can choose and give me what supposed to be the enzyme name. Yes, Balaji. If you raised your hand before, remove it. Yes, Muhammad Abu Ghandar.
Sucrose.
Excellent. Sucrose. Yeah, the disaccharide sucrose. Sucrose
Sucrase
plus water give you what?
is equal to fructose.
Glucose and what?
Fructose and glucose.
And fructose. Excellent,
Fructose.
Muhammad. Yes. Glucose and fructose. What we call the
So
enzyme? The enzyme sucrase.
Sucrase.
Yeah, this is the name of enzyme. So easy to name the enzyme, okay? This is the idea of enzyme. So the name of enzyme is the substrate name, adding ASE to its end, okay? Just modify it by adding ASE. Remember, the substrate are the reactants which are fit with the active site of the enzyme. The active site binding or active site, we called it active site, okay, of the enzyme. Guys, you have to remember, remember important thing. Scientists put a theory for that. They said in the beginning, they said the substrate and the active site are completely fit, or they called it like key and lock. How the key is very fit with the lock, right?
Right.
Mm.
And this theory
Yes.
called lock and key theory. Lock and key theory. Okay? They said the enzyme and the substrate are 100% like each other. The active site and the substrate are 100% fit to each other, okay? But actually, a few years later, they discovered no, it's not 100%. It's similar, but when do you see my camera?
Yeah.
Yeah. When the
Yes.
substrate enter the active site make fitting on the substrate. Okay, again, the substrate enter the active site and the active site make what? Fitting. Okay, so they call this theory induced fit theory. What we called it?
Induced fit theory.
Induced fit
Theory.
theory. This theory says that the substrate enter the active site and then the active site is fitting on the substrate. Okay, but it's still specific. Still, the substrate is or the active site of the enzyme is specific for a substrate. Other substrates, different substrates cannot bind on the enzyme. Okay, is it clear?
Yeah.
Yeah, that's good.
Wait, mister.
Some chemical reactions that make life possible are too slow or have activation energies that are too high to make them practical for living tissue. So these chemical reactions are made possible by catalysts. And we said this catalyst is a substance that speeds up rate of chemical reaction, the time which is needed to convert the reactant into products. Don't forget. Catalysts work by lowering reactions activation energy. What or why are enzymes important to living things? Question, need answer. Why are enzymes important to living things? Yes, yeah, Abdullah Malik. Wait. Guys, make sure that I'll cut much of marks for some people who do not share with us and participate. 10 marks on participation. Okay? This is not accepted from some people. They do not share with us. Only few names open their mic and speak and participate. Others do not share their ideas with us. Hassan Allam, yes. Open your.
Hello?
Yes, hearing you, yes.
Okay. So, hello.
Yeah, the question in front of you. Can you answer it? Why?
Yes, I don't know that much, but enzymes, it allows, okay,
Yep.
it's like it allows the reaction of the occur rate in life, like in the necessary of life.
Yeah. But why is it important?
It's it helps
Okay.
like
Okay. Hassan, because of the time is going to finish, so think about it. After we rejoin, we'll continue. Okay, guys? Yalla, rejoin quickly.
Yes.
Okay. Mr. Can do it.